Every single thing you do, right? Like reading an email or just taking a breath, fighting off a cold, all of it is made possible by trillions of these microscopic cities working in just perfect harmony inside you.
It's astonishing when you really think about it.
It really is. You aren't just one organism, You're like this walking, breathing metropolis.
Yep.
And today, we are going to unpack the architecture of these incredible cellular cities.
Yeah, we've got some great material for this deep dive.
We do. We've spent the past week poring over this fantastic stack of biology revision materials. We're talking brilliant breakdowns from creators like Free Science Lessons, Cognito, and we've been cross-referencing all of that with deep dives from Launchpad Learning and Beverly Biology.
Right, which is such a solid foundation to work from.
Exactly. So the mission today is simple. We're taking you on a microscopic tour of the fundamental unit of life. Okay, let's unpack this. Because to truly understand how you function, we have to start by looking at the completely different zoning laws that govern life on Earth.
Zoning laws is actually the perfect way to look at this, because if you zoom out and look at all life, I mean, the architecture broadly splits into two main styles, right? You've got your eukaryotes and your prokaryotes.
Right.
Animals, plants, fungi, that includes you and me, obviously. We are eukaryotes. Our cells are massive, they're highly complex, and they are meticulously zoned.
I love that visual, the zone city.
Yeah.
Because in a eukaryotic cell, everything has a designated room.
Exactly.
Biologists call these rooms membrane-bound organelles. So the DNA, which is basically the master blueprint of the entire city, it isn't just like lying around in the street.
No, definitely not.
It's locked inside this highly secure double-walled vault called the nucleus. And because eukaryotic DNA is linear-like, these long, unwieldy strings, it's wrapped really tightly around these special spool-like proteins called histones.
To form chromosomes, yeah.
It keeps the blueprints perfectly organized.
It's an incredibly ordered system, and eukaryotes also rely on this massive, heavy-duty machinery to read those blueprints and actually build things. They use what we call ADS ribosomes.
ADS.
Yeah, the ADS is basically just a measurement of how big and dense they are. They're like the industrial-sized protein factories of the cellular world. But then, you have the other architectural style, the prokaryotes.
The bacteria and archaea.
Exactly. If our cells are these sprawling, perfectly zoned cities, a prokaryotic cell is basically a tiny, chaotic, open-plan studio apartment.
An open-plan studio where, like, the cooking, sleeping, and manufacturing are all just happening in the exact same room.
Yeah, it is absolute chaos, but I mean, it's highly efficient chaos. Prokaryotes are vastly smaller than our cells. They have 0 membrane-bound organelles.
No nucleus vault at all.
Nope. Their DNA isn't neatly spooled around histones either. It's just a single, continuous, circular loop of code, just floating completely freely in the cell's fluid, the cytoplasm.
Oh.
And because they're smaller overall, their manufacturing plants are smaller too. They use 7DS ribosomes, so a lighter, more compact version of those protein factories.
But they also have all these rogue survival tools that our heavily regulated cells just don't have. Like, while reading through the sources, the plasmids really caught my attention.
Oh, the plasmids are fascinating.
Right, because prokaryotes have their main DNA loop, but they also keep these little extra mini loops of DNA, the plasmids, just floating around. And these mini loops often contain genetic hacks, like the instructions for antibiotic resistance.
And that's exactly where the open plan architecture really benefits them. They also have this rigid outer defense wall made of a tough chain-link polymer called peptidoglycan, or murine, some people call it.
Oh, yeah.
And many of them even secrete this thick, sticky slime capsule on the outside of that wall.
It's basically stealth tech.
It really is. It makes them incredibly slippery, which physically stops our white blood cells from grabbing and eating them.
And if they want to share those genetic hacks, the antibiotic resistance, they have these little grappling hooks called pili on their surface. They literally latch onto another bacterium and just pass a copy of that resistance plasmid right over.
It's biological piracy.
It is, but okay, I have to stop here and push back on one thing that always completely boggles my mind. Viruses.
Ah, the viruses.
Right, because we talk about the zone cities and the open plan studios,
Yeah.
but viruses cause so much destruction, they replicate, they adapt. Why do scientists so stubbornly refuse to categorize them as living things?
Well, it's a great question, but it comes down to the fundamental machinery of life. Viruses are acellular. They are not cells at all.
Right.
They're just a rogue string of code, either DNA or RNA, wrapped up in a geometric protein shell called a capsid. Sometimes as they exit a host, they'll steal a layer of the host's lipid membrane to cloak themselves, but I mean, that's it.
And they are tiny, right?
Unimaginably tiny, ranging from just 20 to 300 nanometers. What's fascinating here is that despite all the chaos they cause, they are entirely inert on their own.
Because they don't have those ribosomes.
Yeah.
They don't have the 80S or 70S factories. They have literally zero manufacturing capability.
Exactly. Think of a virus as a rogue flash drive containing malicious code. A flash drive just sitting on a desk can't do anything. It has no power, no processor. It absolutely requires a computer to read its code and execute it.
Oh, that's a great analogy.
The virus has to hijack a host cell's enzymes and ribosomes to build more of itself. Without a host, a virus is just a complex, lifeless arrangement of molecules.
That is terrifying, but so brilliant. Okay, so leaving the rogue flash drives behind, let's zoom back into our own zone city, the eukaryotic cell.
Let's do it.
I really want to trace the actual creation of the city's most vital product, which is proteins. Because proteins are everything, right? They're the structural scaffolding, they're the enzymes digesting our food, the transport vehicles, and all of it originates in that secure vault, the nucleus.
The nucleus really is the defining feature of our biology. The wall of this vault, the nuclear envelope, is actually double membrane, meaning it's two separate layers of lipids.
But it's not totally sealed off.
No, it has to communicate with the rest of the cell, so it's dotted with these highly regulated security gates called nuclear pores. Inside, suspended in a fluid called nucleoplasm, is the chromatin, which is that combination of DNA and histone spools we mentioned earlier.
And there's also this really dark, dense mass inside the nucleus called the nucleolus, right?
Yes.
Its entire job is to manufacture ribosomal RNA. It's literally the factory that builds the parts for the protein factories.
Exactly.
But this creates a massive logistical nightmare, doesn't it? Like the master blueprint for every protein is locked inside the nucleus. But the ribosomes, the actual factories, are floating outside in the cytoplasm. And the DNA is way too large and way too precious to ever leave the vault.
It's the classic library problem. You can't just check out the ancient original manuscript and take it to a hazardous construction site.
Right, you ruin it.
So the cell creates a disposable photocopy. This process is called transcription. The cell reads the specific section of DNA it needs and creates a single-stranded copy called messenger RNA, or mRNA.
And because that mRNA photocopy is single-stranded, it's thin enough to just slip right out through those nuclear pores.
Precisely. It slips out into the cytoplasm and meets a ribosome. Now, these ribosomes are actually built in two pieces, a smaller 40S subunit and a larger 60S subunit.
Oh, interesting.
Yeah, and they essentially clamp down on that mRNA tape and start reading the code, translating it into a physical chain of amino acids.
Now, if the cell just needs a simple protein to use locally, it just uses a ribosome that's floating freely in the cytoplasm.
Right.
But if it's building a complex protein meant for export like a hormone or something that needs to be embedded in a membrane, it has to use the heavy machinery. It heads over to the rough endoplasmic reticulum.
The rough ER. It's this massive winding network of flattened membrane sacs called cisternae. We call it rough because its surface is absolutely studded with thousands of ribosomes, giving it this bumpy texture under a microscope.
Right, right.
As the ribosome reads the mRNA, it physically threads the growing protein chain directly into the inner space, the lumen, of the rough ER.
And that's where the folding happens. Because the protein isn't just a straight string, it has to be folded into a very specific 3D shape to actually function.
Exactly. Shape is everything for a protein.
And just down the street from it is the smooth ER. It doesn't have ribosomes, obviously, so it looks smooth. But it's busy synthesizing lipids and carbohydrates and acting as the cell's detox center, breaking down toxins.
So we have our folded protein in the rough ER, but it's raw. It needs processing. So the ER pinches off a piece of its own membrane to create a little protective fluid-filled bubble called a vesicle, trapping the protein inside.
And then that vesicle floats over to the Golgi apparatus.
A Golgi, yes.
Which a lot of textbooks call the Golgi the post office of the cell. But that always felt, I don't know, a little too simple to me. It's more like a highly automated chemical sorting and modification facility. It's another stack of those flattened cisternae sacs, but it actively changes the product.
It really does. It doesn't just slap a stamp on a box.
Yeah.
As the protein moves through the layers of the Golgi, enzymes are physically altering it. They might attach carbohydrate molecules to the protein to create what's called a glycoprotein.
Oh, like a hybrid molecule.
Exactly. And these carbohydrates act like chemical zip codes, ensuring the cell's internal transport network knows exactly where this protein belongs. Finally, the Golgi packages the finished product into a new vesicle, which travels to the cell membrane, fuses with it, and just ejects the protein out into the body.
Exocytosis.
You got it.
It's just a flawless assembly line. Volt to mRNA to rough ER to Golgi to export. And you can actually see this specialization in action, right? Like if you look at beta cells in your pancreas, whose entire job is to manufacture massive amounts of the protein insulin, those cells are absolutely packed with rough ER and Golgi bodies. The architecture of the cell physically adapts to its job.
Form follows function, but I mean, running this incredible manufacturing hub requires a staggering amount of energy. A factory cannot run without a power grid.
Enter the mitochondria, the power plants.
They handle aerobic respiration, they take glucose and oxygen and run them through this series of really complex reactions to generate ATP.
The energy currency.
Right. You can think of ATP as the universal energy currency of the cell. If a protein needs to be built or a muscle needs to flex, the cell pays for it with ATP. And structurally, the mitochondrion is an engineering marvel. It has a smooth outer membrane, but the inner membrane is intricately folded into these structures called cristae.
And those cristae folds are all about maximizing surface area, aren't they?
Exactly.
You want as much physical space as possible for those energy-generating chemical reactions to happen. The fluid inside all those folds is called the matrix. But we also have to talk about the power grid for plants, the chloroplasts. These are the cellular solar farms.
Plants are incredible because they have mitochondria to burn energy, just like us, but they also have chloroplasts to actually create their own fuel from sunlight. Chloroplasts also have a double membrane. Inside, they contain these flattened, fluid-filled sacs called thylakoids.
And that's where the light-to-pleasant reactions happen.
Oh. Yes, because the thylakoids contain chlorophyll, which is the pigment that physically captures solar radiation.
And to pack as much solar-gathering power in as possible, these thylakoids are stacked up like tiny microscopic pancakes into towers called grana.
Grana towers, yes.
And they're all linked together by flat membranes called lamellae, so they can actually share resources.
It's so efficient. The solar energy captured in the grana is then passed out into the surrounding fluid, the stroma. That's where the light-independent reactions occur, taking carbon dioxide from the air and using that solar energy to stitch it together into glucose.
Amazing.
And the plant will often just store that glucose right there in the stroma as massive starch granules for later use.
Okay, wait. I was digging through the notes on mitochondria and chloroplasts, and there's a glaring architectural anomaly here. Eukaryotic cells have ADS ribosomes. They have linear DNA locked in a nucleus. But mitochondria and chloroplasts...
Ah, here we go.
Here's where it gets really interesting. They have their own completely separate loops of circular DNA. And they manufacture their own proteins using 70S ribosomes, the exact same small ribosomes found in bacteria.
Yep.
Why on earth do our power plants look exactly like bacterial cells hiding inside us?
This brings us to one of the most profound, just mind-bending discoveries in modern biology, the endosymbiotic theory. All the structural and genetic evidence points to a really wild conclusion. Hundreds of millions of years ago, mitochondria and chloroplasts were not parts of a cell.
Oh.
They were entirely free-living, independent, prokaryotic bacteria.
Wait, you're saying the power generators keeping my heart beating right now used to be completely independent organisms?
That is exactly what the evidence suggests. The theory is that an ancient, larger predator cell engulfed these smaller, energy-producing bacteria, but for whatever reason, instead of digesting them, they reached a stalemate. A truce, yeah. They formed a symbiotic partnership. The smaller bacteria churned out massive amounts of ATP or glucose for the larger cell, and the larger cell provided a secure, nutrient-rich environment. Over millions of generations, they became entirely codependent. They literally lost the ability to survive alone, but they kept their original bacterial DNA and their 70S ribosomes.
That is genuinely unbelievable. We aren't just one organism. We're like a Russian nesting doll of ancient partnerships.
We really are.
Okay, so we've got the energy grid running. But with all this massive industrial production, the city is obviously generating waste and toxic byproducts.
Yeah.
How does the cell prevent itself from just turning into a toxic dump?
It requires a highly specialized hazmat team. The Golgi apparatus produces these special vesicles called lysosomes. Now, instead of holding proteins for export, lysosomes are packed with incredibly powerful, highly acidic digestive enzymes called proteases.
Right.
They are the cell's waste disposal and recycling centers.
But hold on. If you have a bubble full of powerful acid and tissue-destroying enzymes just, like, floating around inside the cell, isn't that incredibly dangerous? If a lysosome pops, doesn't it melt the entire cell?
It would, except for a brilliant evolutionary safety mechanism. Those digestive enzymes are evolved to only function in a highly acidic environment. The lysosome actively pumps protons into itself to keep its internal pH very low.
Oh, I see.
So if a lysosome accidentally ruptures and spills its contents into the cytoplasm, the neutral pH of the cell immediately deactivates the enzymes. The acid is diluted, the enzymes shut down, and the cell survives.
A built-in kill switch.
Yeah.
That's incredible. So they use these safeguarded enzymes for phagocytosis-like. When a white blood cell swallows an invading bacteria, traps it in a bubble, and then the lysosomes fuse with the bubble to digest the invader alive.
Exactly.
They also do autolysis, where they seek out old, broken-down organelles, like maybe a worn-out mitochondria, and break it down into raw materials so the cell can recycle the parts.
They are ruthless, incredibly efficient recyclers. Now, while we're talking about managing fluids and waste, we have to contrast how animal and plant cells handle storage. Animal cells might have small temporary vacuoles, like the bubble used to trap that bacteria we just talked about.
Yeah.
But plant cells have a massive, permanent central vacuole. I mean, it can take up to 80% of the cell's entire volume.
It's basically a giant internal water tower, and it's surrounded by its own specialized membrane called the tonoplast, which rigorously controls what goes in and out.
Right.
The fluid inside is called cell sap.
Right.
It's this dense mixture of dissolved sugars, mineral salts, amino acids, and sometimes even vibrant colored pigments to attract bees or toxic poisons to stop caterpillars from eating the leaves.
But the real genius of the vacuole is structural. Because that cell sap is so densely packed with sugars and salts, it creates a powerful osmotic gradient. It has a very low water potential compared to the fluid outside the cell. As a result, water is constantly trying to rush into the vacuole to balance it out.
And as that water rushes in, the vacuole inflates like a balloon, right, pushing outward against the rest of the cell. Biologists call this outward hydrostatic pressure turgidity. It's exactly what keeps a plant stem firm and upright.
Exactly.
When a plant doesn't get enough water, the vacuole shrink, the pressure drops, and the whole plant wilts.
But this raises a major mechanical problem. If you have a massive water tower constantly inflating and creating intense outward pressure, how does the cell actually maintain its shape? How does anything move around in this pressurized environment?
Right. The city needs infrastructure.
Yeah.
It needs scaffolding and roads. In a cell, this is the cytoskeleton.
Hmm.
And I used to just picture this as rigid steel beams holding up a building. But reading these sources, it's so much cooler. It's a dynamic, self-assembling protein monorail system.
It's an incredibly active network. The cytoskeleton has 3 main layers. The narrowest fibers are the microfilaments, which are made of a contractile protein called actin.
Actin. That's the exact same protein that allows our huge muscle fibers to physically contract.
It is.
But even in a microscopic single cell, they cause movement. Like when a cell divides in half, the actin microfilaments form a ring around the middle and literally pull inward like a drawstring on a bag, pinching the cell into 2 new cells.
Cytokinesis, yep. Then you have the intermediate fibers. These are more like the tension cables of a suspension bridge. Think about your skin cells. They are constantly being pulled, stretched, and compressed. The intermediate fibers anchor the organelles in place and absorb that mechanical stress so that the cell doesn't literally tear apart when you bend your elbow.
Wow. And the largest structures are the microtubules, made of a protein called tubulin. These are the literal highways of the cell. Earlier, we talked about a vesicle traveling from the rough ER to the Golgi. It doesn't just lazily float through the fluid, it is physically carried.
Yeah, this part is amazing.
Special motor proteins grab the vesicle and literally walk it, step by step, down the microtubule highway.
It is such a stunning visual to imagine. And microtubules are also responsible for moving chromosomes. When a cell divides, it has to drag its copied DNA to opposite sides of the cell. The microtubules assemble into these long cables called spindle fibers to essentially reel the DNA in. And the structures that organize this entire process are called centrioles.
Centrioles are these little cylinders made entirely of microtubules. 2 of them sit at right angles to each other near the nucleus, forming a centrosome. And they act as the central anchor point for building all those spindle fibers.
Though, nature always has exceptions. Interestingly, flowering plants completely lack centrioles, but still manage to organize their spindle fibers perfectly fine.
Of course they do.
However, in animal cells, centrioles have another really vital role. They form the anchor base for cilia and flagella.
These are the structures that stick out of the cell to create movement. And I am completely obsessed with their internal architecture.
It's so elegant.
If you take a cilium, slice it in half, and look down the tube, it is always constructed the exact same way. 9 pairs of microtubules arranged in a perfect outer ring, with 2 single microtubules right in the center. The 9+2 structure. It's like a universal architectural standard for cellular movement.
It's a beautiful geometry, and they move by using ATP energy to make those outer pairs of microtubules actually slide past one another. Because they are anchored at the base, that sliding action forces the entire structure to physically bend and whip.
Oh, that makes sense.
Cilia are short, and a cell will have thousands of them beating in unison. Flagella are long and whip-like the tail in a human sperm cell propelling it forward.
But cilia aren't just for swimming. They act like microscopic conveyor belts.
Yeah, if we connect this to the bigger picture, think about your trachea. The lining of your windpipe is covered in millions of these beating cilia. They constantly walked upward in a synchronized ways, pushing mucus and trapped dust out of your lungs so you can swallow or cough it out.
Right.
If those cilia fail, say paralyzed by chemicals and cigarette smoke, the lungs can't clear the debris, which cascades into chronic respiratory disease. Cilia also line the fallopian tubes to waft the egg to the uterus. It's astonishing how a microscopic 9+2 arrangement of tubes dictates human breathing and reproduction.
It's breathtaking. But speaking of architecture and tension, we have to go back to the plant cell's water tower really quickly. We established that the vacuole creates immense outward pressure. Our animal cells are just wrapped in a soft lipid membrane. If we took on that much water pressure, we'd pop like an overfilled balloon.
Exactly. So plant cells construct a rigid physical box around their flexible membrane, the cell wall. In plants, this wall is made of cellulose.
Right.
Cellulose is a complex polymer. It's made of long, straight chains of a sugar called beta-glucose. Because of how the molecules are shaped, these chains form countless hydrogen bonds with the chains next to them, creating these incredibly strong, thick cables called microfibrils.
And the plant takes these strong microfibril cables and weaves them into a crisscrossing mesh. It's like biological Kevlar.
That's a great way to put it.
It's porous enough to let water and nutrients flow right through, but mechanically it refuses to stretch. So when the vacuole fills with water and pushes the soft membrane against the wall pushes back. That equalized tension is what allows a 300-foot redwood tree to stand upright without a single bone in its body.
And to keep all those individual brick-like cells from falling apart, plants secrete a layer between the cells called the middle lamella. It's really rich in calcium and acts as a biological cement, gluing the adjacent cell walls together so the entire tissue acts as one solid unit.
And fungi do something similar, right?
They do to resist pressure, but they build their walls out of a completely different polymer called chitin, which happens to be the exact same tough material beetles use for their exoskeletons.
It is truly a feat of engineering at a scale our brains can barely even comprehend. So what does this all mean for you, the listener? How does this change the way you interact with the world? The next time you take a bite of a crisp piece of celery, you hear that loud crunch, you aren't just chewing a vegetable.
No, you're not.
You are physically fracturing billions of woven cellulose microfibrils. When you flex your bicep, you are sliding millions of actin proteins past each other, powered by the chemical ATP currency generated along the folded cristae of your mitochondria.
It connects every mundane action we take to a highly ordered, microscopic universe operating just beneath our skin, and it leaves us with something quite profound to consider, I think.
Yeah.
If the endosymbiotic theory holds true, those power-generating mitochondria beating inside your muscle cells right now are the direct descendants of ancient, independent organisms that moved into our cellular ancestors and simply never left.
Meaning we aren't just built of cities, we are built of ancient partnerships.
It makes you wonder, are we truly single, solitary individuals? Or are human beings actually walking, talking, highly cooperative ecosystems? Something to ponder the next time you look in the mirror.